Since Mendel knew nothing of the chemical basis for inheritance, he had to work very carefully and examine only the external appearance of the plants. He compared what we call today the phenotype, which is the outward expression of the genes. Mendel began his studies on a total of 34 distinct varieties of green pea. For 2 consecutive years, he grew each of these varieties in separate plots, to each for their purity and to assess the stability of their characters. That is, how much the expression of characters was influenced by the environment. Finally, he selected 22 of these varieties for hybridization. Each of these varieties differed from each other with respect to one or more characters. A character is any morphological, anatomical, biochemical, or behavioural feature of an organism. For example, plant height, seed shape, seed color, etc. Mendel selected 7 characters for his study, and each of these characteristics had 2 alternate or contrasting forms. The characteristics include height, which may be tall or short, pod shape, which can be inflated or constricted, seed shape, which can be round or wrinkled, seed color, that can be green or yellow, and so on. Mendel crossed varieties differing from one pair of contrasting characters. Varieties differing from one another for one or more characters and used in hybridization program are known as parents. Crossing or hybridization consists of placing active pollen grains from a flower of male parent onto the stigma of emasculated flowers of the female parent. Emasculation is the removal of all the immature anthers of a flower to prevent self-pollination. The progeny obtained by crossing are known as hybrid or F1. F1 stands for first flyle or progeny generation. Self-pollination in F1 produces F2, while that in F2 generates F3, and so on. In one of his cross, Mendel crossed a variety tall plants with short plants. The plants resulting from hybridization in F1 were all tall. The F2 plants were of 2 kinds. On an average, 3 out of 4 plants were tall, and 1 out of every 4 plants were short. Mendel raised the F2 plants, allowed them to self-pollinate, and studied the height of plants produced by each F2 plant. He compared these findings with the height of the plant that produced the F2 in question. He found that all the plants from short F2 plants produced only short F3 plants. But F3 plants raised from F2 tall were of 2 types. On an average, 1/3 of such plants produced only tall F3 plants, while 2/3 of such plants produced both tall and short F3 plants in the ratio 3:1. Mendel further noted that the results from reciprocal crosses were identical. Since garden peas are sexual organisms, each cross can be performed in 2 ways. The 2 crosses in which the same 2 parents are involved, but the strain which serves as the male parent in one cross is used as the female parent in the other, and vice versa, is called a reciprocal cross. A cross between the parents differing for a single character is termed as monohybrid cross, while those between parents differing for 2 and 3 characters is known as dihybrid and trihybrid cross respectively. Results obtained from monohybrid crosses for the remaining 6 characters were identical with those for plant height. He soon figures out what was happening. Since each character he was studying had 2 forms, now let me deliberate on law of segregation. The results of Mendel's initial breeding experiment formed the basis of his first law of heredity that I have already told you, the law of segregation. The concept of segregation identified as Mendel's principle can be phrased once again as the 2 alleles present in the F1 are able to separate and pass into different gametes in their original form, producing 2 different types of gametes in equal frequencies. This principle is a statement about genetic transmission. An allele is transmitted faithfully to the next generation even if it is present with a different allele in a heterozygote. The phenomenon of segregation can be easily explained on the basis of behavior of homologous chromosomes during meiosis. As a consequence of segregation, the 2 alleles of genes separate and go on into different gametes. Therefore, the biological basis for this phenomenon is the separation of homologous chromosomes during meiosis, as the alleles are located in identical positions in the homologous chromosomes. As far as the time of segregation is concerned, there are 2 cases. Number 1, when there is no crossing over between a gene and the centromere of the chromosome carrying this gene. In that case, the alleles of the gene segregate during the first meiotic division, which is the reductional division in such case. Case second, when there is a crossing over between the gene and the centromere. In that case, the 2 alleles of the gene segregate during second meiotic division, which is the reductional division in such cases. Mendel concluded that each one was controlled by a single factor. Today, we call these factors as genes. Therefore, each gene has 2 alternate forms. These forms are known as alleles. One form is dominant while the other is recessive. When the dominant form of a gene is present, it covers up or masks the recessive form. At first, Mendel found the above results puzzling, but he had a logical mind and was good at mathematics. Mendel also realized that in every generation, each offspring inherits one form of the gene for character from one parent and another form of the gene from the other parent. So, if one of Mendel's P inherited 2 dominant forms of the plant height gene, that is homozygous dominant, it will give rise to a tall plant. An offspring having 2 copies of the same allele, such an organism is homozygous. And the situation is known as homozygosity. If another P inherited one dominant form and one recessive form, that is, the condition is heterozygous, it would give rise to tall plants because the dominant form would mask the expression of the recessive form. When an individual has 2 differed alleles of the same gene, it is referred to as heterozygous, and the condition is termed as heterozygosity. Mendel used the terms pure and hybrid instead of homozygous and heterozygous respectively. But if a third P inherited 2 copies of the recessive form, that the condition is homozygous recessive, it would give rise to short plants. In Mendel's second generation experiments, these combinations were what produced the characteristic 3:1 ratio of tall to short plants. Mendel used symbols to represent the heredity factors that he postulated, which was a methodological breakthrough. With symbols, he could describe heredity phenomena clearly and concisely, and he could analyze the results of crosses mathematically. He could even make predictions about the outcome of future crosses. Although the practice of using symbols to analyze genetic problems has been much refined since Mendel's time, the basic principle remained the same. The symbols stand for genes, or more precisely we can say for their alleles, and they are manipulated according to the rules of inheritance that Mendel discovered. These manipulations are the essence of formal genetic analysis. Dominant alleles are represented by capital letters, whereas recessive alleles are represented by lower case letters. Dear students, after 8 years of hard work, Mendel has discovered several foundational principles that govern heredity. They are: number 1, law of unit characters, which states that each character is controlled by an independent factor as a unit. This law is no more acceptable now, as we have instances of pleiotropy. Number 2 law, that is the law of segregation, which states that the factors are separated during gamete formation. Now this law is called as the first law of Mendel. Third law, that is law of dominance. As per this law, if 2 contrasting characters were brought together in a cross, one of them expresses itself. This law also is not true in all cases, as we have instances of incomplete dominance, codominance, and so on. Fourth, that is the law of independent assortment. According to this law, factors for different characters behave independently. This is called now the second law. Mendel presented the results of the experiment at meetings of the Natural Science Society in Brno in 1865. A year later, he published a formal paper with his results and conclusions about how characters are inherited. He sent reprints of his paper to prominent scientists throughout Europe. Unfortunately, Mendel's paper was largely ignored. There were several possible reasons for this. First, Mendel's result did not match those of other scientists who were also using plants to try to study heredity. Second, other scientists of that time were not using math to explain observations in the natural world. They did not understand Mendel's ratios. And third, most scientists of Mendel's time focused on things they could actually see in their research. They wanted to know exactly what Mendel's factors were, what did they look like, where they are located. No one, not even Mendel, could answer these questions. Later, Mendel became more and more involved with the work of monastery, and he was made abbot in 1868. But he still found time to continue his studies on honeybees, some other plants, and on climatology. Johann Mendel died on 6th January 1884 at the age of 62 of a kidney disorder. By that time, his paper had been gathering dust in libraries all over the Europe for 2 decades. 16 years after his demise, 3 scientists working independently of each other, De Vries in Holland, Corens in Germany, Schmarck in Australia, arrived at the same conclusion as those of Mendel. These 3 scientists rediscovered the forgotten paper by Mendel and proclaimed to the world its importance in 1900. After this rediscovery, Mendel's ideas quickly gained acceptance, especially through the promotional efforts of the British biologist William Bateson. This champion of Mendel's discoveries coined a new term to describe the study of heredity, genetics, derived from a Greek word meaning to generate. Do you ever wonder why you are the only in your family with your grandfather's nose? The explanation to this, that is the way in which traits are passed on from one generation to the next and sometimes 6th generation, was first explained by Johann Mendel, a monk at the monastery at St. Thomas in the town of Brunn in the Czech Republic. Mendel did not discover these foundational principles of inheritance by studying human beings, but by studying the common pea plant. He even tried some experiments with honeybees. His greatest success, however, was with peas. He conducted all his experiments within the kitchen garden of his house with the help of his own resources. He began to collect pea seeds for his experiment in 1857 from commercial growers all over the Europe. Mendel developed a systematic way of understanding heredity. He conducted all his experiments by using very good scientific technique. He applied the scientific method combined with mathematical analysis to propose foundational principles of inheritance that described the transmission of genetic traits before anyone knew genes existed. Today, whether you are talking about pea plants or human beings, genetic traits that follow the rules of inheritance that Mendel that time proposed are called Mendelian laws. Mendel was curious about how traits were transferred from one generation to the next, so he set out to understand the principle of heredity in mid-1860s. Mendel chose the garden pea as his experimental material because it offered several advantages as an experimental material of which Mendel was well aware. These advantages are: number 1, they are very productive. That means they produce many seeds. Number 2, they have short life cycle. That is, they produce many generations in a short time. Number 3, they can be easily cross-pollinated due to flower structure. Number 4, several characters of the pea plant have 2 contrasting forms which can be easily distinguishable from each other. For example, tall versus short plants. Number 5, they are easily grown in experimental garden or in pots in a greenhouse. Number 6, one of the peculiarity of pea reproduction is that the petals of the flower close down tightly, preventing pollen grains from entering or leaving. This enforces a system of self-fertilization in which the male and female gametes from the same flower unite with each other to produce seeds. As a result, individual pea strains are highly inbred, displaying little, if any, genetic variation from one generation to the next. Because of this uniformity, we say that such strains are true breeding, which means that plants produce only progeny like themselves when allowed to self pollinate normally.